Hi, my name is Brianna Wolotka, and in my presentation, I will be talking about the behavior of pill bugs with the concept of taxis. And before I actually start this presentation, I would like to apologize to whoever's listening. I am feeling a little bit under the weather. It's nothing serious. I just want to let you know that if I sound weird or if I cough or sneeze, which is gross, I am sorry, and I will try to refrain from doing that. But here's my presentation. So, what exactly are pill bugs? Pill bugs are small nocturnal isopods, which is a non-type of arthropod, also known as a terrestrial crustacean. They inhabit areas that are dark and moist, and you can usually find them under rocks, leaves, or fallen trees/logs. We mostly know them as roly-polies rather than pill bugs. So knowing their natural habitat, I decided to focus my experiment on the effect of the light on the movement of pill bugs with taxis. So saying that, what does taxis actually mean? Our lab book describes it as one of two oriental behaviors that, in response to a stimulus, such as food or, in my study's case, light, causing the organism to move directly away or towards the stimulus. If the organism responds to the stimulus by moving away, it is called a negative effect, and if the organisms respond by moving towards the stimulus, it is called a positive effect. So taxis is actually commonly mixed up with the second oriental behavior, which is kinesis, because they are so similar. So kinesis is like taxis in a way that the organisms respond to a stimulus, but the movements will be non-directional, meaning that the organism will not move towards or away from the stimulus, rather they will move at a faster or slower pace. So, why is this experiment important to us and biologists and any others who would like to study this? This can help us understand how the pill bugs use their senses to react to their surroundings when put in an unexpected situation, such as exposure to light, if the light does affect them. This can also explain how the organisms are able to find their environments with barriers such as a light present. So for this experiment, my hypothesis was that if I gave pill bugs a covered moist habitat and an uncovered dry habitat, then when a light is shined on the two habitats provided, they will respond by going into the moist covered habitat. So if I shine the light on the two habitats provided, it would have a negative taxis effect on the bugs. My predictions, similar to my hypothesis for this study, is that the light will have a negative effect, causing the pill bugs to move away from the light to the covered moist habitat provided. So to actually conduct this experiment, I have decided to do one experiment, which is the taxis effect on the movement of pill bugs. The materials that I will need for this experiment are very specific 12 pill bugs, an empty tank, and some sort of blackout material. And I'm not really too specific, whether it be black curtains, black construction paper. I just would have needed something to make half of the tank pitch black, so that way I can make it as close to their natural environment as possible. I would also need a divider to put between the covered and uncovered tank with a opening in between so that they could move freely between the two sides. I would need soil for the covered side and a flashlight or some sort of other source of light, whether it be a heat lamp or lights from a room, and a stopwatch. So due to my circumstances, I was not actually able to perform this experiment, but how I would have done my experiment is that I would have first put them in a control where I will place the 12 pill bugs into the covered side of the tank and allow them to just kind of get accommodated and stay there for about 30 seconds. And then once that time is up, I will carefully pick the pill bugs out to make sure that, none of them get squished and mess up my experiment, and place them into the uncovered empty side of the tank, where I will then begin the experiment. I will turn the light source on for 6 minutes, and every 30 seconds I will record how many pill bugs are in each side of the tank, and I will record this data into a chart or a table. I want to apologize for this slide. The 6 minute timestamp, is a little hard to read. I didn't want to just put it on a separate slide. I didn't want it to make it just kind of seem boring or just kind of put there to create more room. It's not. I just didn't want it to, I wanted it to be all in the same place, so I'm sorry. Due to the coronavirus and the circumstance that we are in, I was unable to complete this experiment myself, as I've said, so my results will be as I predicted them to be, but I would also like to note that I used lab results from another experiment that I found online, similar to my own, just to kind of make this experiment as more accurate as possible, rather than saying, yeah, all 12 pill bugs went into the covered area, because that might not be true if I was actually able to perform this experiment. But what we can assume from this data is that there could have been a few strays or errors that could have happened, such as, in the worst case, a pill bug passing away due to being squashed or drying out and dying. It happens, but again, very extreme. But it could be on my part, a human error. I could have miscounted or put it in the wrong slot for a different time, causing my data to be messed up. But again, we can assume from this that the pill bugs did respond negatively towards the light, causing them to go into the covered environment. So overall, my data does correlate with my hypothesis and predictions, but my data is not factual. As I did say, I did not conduct this experiment, so this is just what I think, not what actually could have happened. So now that we know that there is a negative effect of the light in response to the movement of pill bugs, biologists can test how other isopods react to the light and why it impacts their movement. So while I was doing this, I was thinking of a couple other experiments that could have been done, which I would have thought would have been cool. One being that you can perform the same experiment but change the colors out to like 4 different colors. So red, blue, green, and just kind of like a natural like white color, to see if it has the same effect on the pill bugs. Like maybe red or blue doesn't make them have a negative effect because it's more dark and it creates a dark environment, whereas like the green and the white natural color might cause a negative effect. I was also thinking about you could do another experiment similar to this one, again, where you can change the type of environments given to sand or something that they aren't usually used to. So I actually looked that one up because I thought that would be really cool if I would, be able to change it to sand on one side and just kind of just have a different environment on the other side. And someone actually did complete this experiment, but not exactly how I would have wanted to do it. They still kept the moist soil on the other side, but they did put sand on the dry, uncovered side and shine the light on them for about 7 minutes. And so what they found out was that it had the same effect. They did not like the sand at all. They actually, a lot more pill bugs went into the moist covered side rather than being in the sand, due to the fact that they're not used to it. So yeah. So actually, I forgot to mention this, but there's a fun fact about them. I mentioned earlier that they might dry out, meaning that they need water to survive. And so actually, pillbugs, the reason they roll up sometimes, besides to protect themselves from predators, is that to make sure they do not dry out, because they actually need water for their lungs, so to function properly. So if they feel like they are going to actually dry out and die, they will roll themselves up into a ball to preserve the moisture they have left in their bodies, to make sure that they do not die. And that is the end of my experiment/presentation. Thank you for watching and have a great day.